So my name is Diogo Ribeiro. I'm a PhD student at Université Laval. And basically today I'm going to talk about gold mobility and metal mobility in metasedimentary belts and its implications for gold endowment. So it's usually generally accepted that the fluid is sourced by that is involved in orogenic gold deposits is usually sourced by the metamorphism and dehydration of supercrustal rocks, such as metasedimentary rocks. In the Phanerozoic, metasedimentary rocks are usually considered a suitable source of gold and other metals for orogenic gold deposits, as it has been showed in previous works from Pitcairn and others. But in the Archean, they've been mostly disregarded because of their low abundance in the greenstone belts, which usually host the orogenic gold deposits. However, in the superior craton, we have highly metamorphosed and extensive metasedimentary belts, which are adjacent to less metamorphosed greenstone belts, which host variable amounts of gold. Pitcairn and others already started this work, and they've seen that there is a systematic decrease of gold, arsenic, and antimony with increasing metamorphic grades, meaning that these metals are being mobilized during prograde metamorphism. However, they do not have constraints regarding PT conditions and the key metamorphic reactions leading to the release of these elements. So that's what the project aims to do, to see whether the Quetico and the Pontiac were suitable sources of fluid ligands and especially metals. For that, we're going to track gold mobility during prograde metamorphism. So basically, I'm responsible for whole rock geochemistry, especially ultra-low detection limit analysis for gold. I'm combining that with in situ laser ablation data in sulfides. And that will be combined with the PT constraints coming from Adrian and Isaac working in the Quetico and the Pontiac. Regarding my project specifically, I'm working in 3 different areas with contrasting gold endowments. The first one is Juan Oranda in the Pontiac, which is considered to be the highly endowed area, adjacent to the Abitibi Gold Camp. Then we have Geraldton, which is adjacent to the Beermore-Geraldton Gold Camp, which is considered here to be moderately endowed, and then Thunder Bay, which has no gold occurrences around. And we aim to compare these areas. So I went to the field, I sampled across all metamorphic zones, aiming for spatial and protholite representativity. I got a suite of samples, mainly metasedimentary rocks and minor mafic volcanic rocks. And I selected some for whole rock geochemistry, and that's the data we're going to look at right now. So this is the data for the 3 areas for arsenic and antimony, and what we can see right away is that the initial concentration of arsenic and antimony is identical in the 3 areas, and that there is a systematic decrease of these elements during prograde metamorphism. So they're being mobilized. But then when we look, will we look? Yeah, we will. Okay. When we look at gold, we don't see, even though we see a decrease of gold contents with increasing metamorphic grade in Juan Oranda and Geraldton, we don't see the same in Thunder Bay. So for some reason, gold is not being released. And to do that, to understand this, we have to look at the minerals in the rock and look at where gold is. This is generally the mineral composition of a low-grade metasedimentary rock. And after some tests, and as we predicted, the gold is mainly hosted in sulfides. So before I go into the in situ data, I will just talk a little bit about the evolution, the textural evolution of these sulfides. So in general, we see a common evolution in the 3 areas, starting with the first generation of pyrite that eventually breaks down to pyrrhotite with increasing metamorphic grade. This breakdown is usually characterized by the presence of transitional sulfide phases that have composition in between pyrite and pyrrhotite. And then at higher grades, a second generation of pyrite, usually hydral, starts growing inside pyrrhotite and eventually consumes it completely. So generally this is what happens, and now we're going to look at the in situ data. We'll start with Juan Oranda, and what we can see is that pyrite 1 is the main host of these three metals. And the content of these metals tend to decrease in pyrite 1 as it recrystallizes during increasing metamorphic grade. And then when we compare with pyrrhotite, we can see that it holds, the concentrations are very low, meaning that these elements were released, as we've seen in the whole rock data. In Geraldton, we see the same thing. Pyrite 1 is the main host of gold, arsenic, and antimony, and pyrrhotite has very low concentrations in these metals. So the pyrite 1 to pyrrhotite transition is the main reaction controlling the liberation of these elements from the metasediments. Then when we look at Thunder Bay, what we see is that even though, so pyrite 1 is still the main host of these elements, but even though we see a decrease of arsenic and antimony, for some reason, gold is not being released from pyrite 1 while it recrystallizes. And that's basically the issue. We don't see gold depletion or as strong of a gold depletion in the whole rock data with increasing metamorphic rate, and pyrite, the recrystallization of pyrite doesn't seem to be liberating gold by itself. So when we look at the textural or the sulfide evolution of each area, we can see that Thunder Bay is different for two main reasons. The first one is that pyrite 1 is more abundant, and it stays stable, remains stable at higher grades, and there's a general low abundance of pyrrhotite. If we look at the model proportions of pyrite 1 with increasing metamorphic rate, comparing the three areas, what we can see right away is that by the biotite zone, in Juan Oranda, there's much less pyrite 1 compared to the other areas. So the breakdown of pyrite 1 to pyrrhotite is pretty abrupt. It's already below 40% and then drops to under 10% and remains like that until the sillimanite zone. In Geraldton, the breakdown is more progressive, but after the staurolite zone, it's usually always below 10%. And then in Thunder Bay, even though we have a decrease up to the garnet zone, the model proportions tend to remain higher. Here over 40% in the staurolite zone and over 20% in the melt zone. So this can explain why gold is not being mobilized from metasedimentary rocks in Thunder Bay. Another thing that adds to this is that when we look at the transition textures, instead of having these intermediate sulfide phases, what we usually see is more a pyrite 1 that is broken and sealed by pyrrhotite. So the textures are also different. So basically, in conclusion, the whole rock data, even though it shows identical arsenic, gold, and antimony concentrations in low rock in the three areas, there is a systematic decrease of arsenic and antimony in the three areas. In Thunder Bay, gold is not being mobilized. Then when we look to the sulfides, we see that pyrite 1 is the main host of these elements, that there are two main reactions, pyrite 1 to pyrrhotite, followed by pyrrhotite to pyrite 2, and that the pyrite to pyrrhotite transition is the main reaction leading to the release of these elements. In Thunder Bay, gold liberation was precluded due to the incomplete nature of this reaction. So what we see is that there is a spatial correlation between gold mobilization in the metasedimentary belts and gold endowment in the greenstone belts. So this is the first part of my PhD. The paper has been already submitted at the end of January, so it's under review. And now I'm going to show you the other two parts of my PhD. So the second part is to go a bit deeper on why gold is not being mobilized from the metasediments in Thunder Bay. The first question is, what controls the relative stability of pyrite and pyrrhotite? And the second one is, how is gold hosted in pyrite? So on to the first question, when we look at the titanium-bearing phases, what we usually see is that in low-grade rocks, titanite and rutile are stable, and then after that, in higher grades, ilmenite is very abundant, and it's the main titanium-bearing phase. And by the sillimanite zone, the titanium-bearing phases tend to disappear. But in Thunder Bay, this is not as clear. Titanite and rutile are found in higher grades, and also there's quite a lot of rocks in low grades that have no oxides at all. So the question is whether these phases are controlling the oxygen fugacity and sulfur fugacity in the rocks, and and if it's the control that leads to the incomplete breakdown of pyrite 1. So these diagrams show the amount of pyrite 1, pyrrhotite, and pyrite 2 in each rock. Each point is a sample for the 3 areas. And in yellow and green, we have rutile and titanite as the dominant titanium-bearing phase. And then in blue, in different shades, the lighter colors are also ilmenite, but ilmenite is very rare or not as abundant. And then the red dots are samples with no oxides at all. So what we see is that the samples that are dominated by pyrite 1 tend to have titanite and rutile as the main titanium-bearing phase. And then when these rocks are eventually dominated by pyrrhotite, ilmenite is the main titanium-bearing phase stable. And then when we look at the rocks that have more pyrite 2, these ones tend to have either very low amounts of ilmenite or no oxides at all. So it seems to me that the presence of ilmenite is maybe required for the stabilization of pyrrhotite, but that's something we're still looking at. The second question was about how gold is hosted in pyrite 1. I'm going to show you some examples, an example from the in situ data I have in pyrite 1 from Rwanda. And basically, for example, when we correlate gold with other, or when we plot gold against other elements within pyrite 1, and we see a correlation, that can tell us that gold may be hosted in nano-inclusions of a different mineral within pyrite 1. And what we see is that there is a good correlation of gold with antimony, lead, copper, and bismuth in pyrite 1 from the biotite zone, and then that correlation kind of fades with increasing metamorphic rate. If we look just at those samples from the biotite zone, and we see the correlation of antimony with lead, copper, and bismuth, we see that there is actually a correlation close to 1-to-1, and this may mean that gold in this pyrite is related to sulfur salts that bear these elements. But when we include the other metamorphic zones, we see that antimony still correlates pretty well with lead, but the correlation with the other 3 elements kind of vanishes, and especially gold, the correlation stops. So basically this means that while pyrite is recrystallizing, the chemistry of these sulfur salts may be changing, and more importantly, they seem to be releasing gold. But we're still working on this. When we look at chemical data and laser ablation maps, we see this correlation of antimony, lead, and bismuth. Here we see other correlations like arsenic, cobalt, and nickel, for which I have no explanation yet, but the data seems to agree. And then when we look at, when we compare Rwanda with Geraldton and Thunder Bay, in Geraldton, when we plot antimony against the other metals, we see also a good correlation, especially with lead, throughout the different metamorphic zones. And these values are in brackets because I have just 6 spots here, so they have to be taken with a grain of salt. But then also a bit of correlation with gold that fades away with increasing metamorphic rate. And then in Thunder Bay, we see no correlation at all. I did some tests, and gold correlates just with, has only good correlation with silver. So this probably means that the gold is also hosted in a different manner in pyrite 1 from Thunder Bay, besides the fact that pyrite 1 itself is not breaking down efficiently. The third part of the PhD will be focusing on whole rock sulfur isotopes. We decided to do this because Cap33 signatures can be used as a tracer, and we wanted to compare the signatures of the sediments with the same signatures in sulfides from nearby orogenic gold deposits. And the data shows that our samples follow the Archean array. This means that the sulfides were precipitated probably from Archean seawater. We don't see any correlation with metamorphic grade. It seems like it doesn't really change the signatures. There seems to be a correlation regarding the dominant main sulfide, but I'm not too sure about this. It seems like that the pyrite 1 dominated samples tend to have lower Cap33, and the pyrite 2 dominated samples have higher Cap33, but this could also be just a difference within the sedimentary sequence, so not too sure yet. And then when we look at Juanoranda specifically, and we compare with the data from Herzog and others, 2024, they analyzed Cap33 and sulfides from Val d'Or. We see that our span of signatures does not match theirs, although it does match Canadian Malartic, which is hosted in the Pontiac, so that makes sense. And so this concludes that even though Juanoranda, the Pontiac sediments may be a source of gold, they're probably not a source of sulfur. They may be locally sourced, I don't know from where. And yeah, that's it. Thank you.